All right, my group stuff is on stem cell and its role in repairing the heart. How hard can repairing the heart be? The answer is very hard. So we're going to go over, there we go. A couple of things. One is the study that we're going off of. Obviously, that's stem cells in the news article. We had to find an actual study. And then we're going to talk about what stem cells they use, some of the techniques they use, why I didn't like this study, and what we hope to build off of it and do differently. So first, let's get into the study. Our study was randomized trial of targeted transendocardial mesenchymal precursor cells and therapy with patients with heart failure. So this study had two main goals, which are its primary endpoint and secondary endpoint. So its primary endpoint was to reduce major heart events. So those are things like strokes, hospitalizations, deaths, heart attack, heart failure, fibrosis, just like any general affliction with the heart. They wanted to reduce the amount of times patients would go to the hospital within a year. And they also wanted to see a reduction of these events across the board. So for every single individual event, they cataloged all of them, whether it was death, any sort of stroke, whether it was ischemic or not, and then what type of heart failure it was, all that kind of stuff. Their kind of proposed method was that they were going to inject stem cells into the heart, and then they were going to be activated by the inflammatory signals that the heart sends out when its muscles are failing, and then they would exert a different signal that would repair the heart by promoting these things called neovascularization, which is basically mini veins within your heart muscles, as well as stop the inflammation and make it to where there's a less chance of strokes and less chances of heart attacks. So let's talk about the stem cells they use. They use allogeneic mesenchymal precursor cells. So break it down, allogeneic is from someone else. These are from donor cells. It was 3 healthy adult donors, and they are multipotent cells derived from bone marrow. So the way they separated them out is that they were targeting these STRO3 surface proteins. That means that it is the type of stem cell that they want to have. And they basically had a monoclonal antibody, which is a highly specific marker, with a magnet on the other end. And they just kind of bound the cells to the magnet and then separated out all the cells that were not magnetized, basically fell through, and these are the bad ones, and then only the good ones stayed up here. So that way they could kind of have the cells they want to, and then they were expanded using proprietary techniques. They didn't tell us what they used to expand them, but they were cultured and frozen so they could have enough cells for this study. This study did about 500 people coming from 3 healthy adult donors. So you need to kind of culture a lot of stem cells there. This is the methods. So they introduced the stem cells transendocardially, which means that a catheter was inserted through one of the valves of the heart and then into the muscle lining of the inside of the cell. This is a very invasive procedure. It differs because a lot of patients will also get injected into the vein or injected into the outside of the heart. This is inside the heart, and they did about 15 injections into the muscle of the myocardial muscle from the inside. And then the hope was that these MPCs respond to the inflammatory signals sent out by this M1 response cell right here, and then these signals would make the MPC start secreting these signals, the PGE2 and the IDO, which would change M1 into M2. M2 inflammatory response cells they are anti-inflammatory. They also promote mostly healing rather than promoting things like apoptosis and inflammation. So this would do 2 things. One, it would create more neoveins, so smaller veins to also help stop inflammation. And 2, it would change the inflammatory response to a healing response instead of the immune response cells also causing inflammation. And then they've kind of tracked patients for 2.5 to 5 years to seeing how many heart failure events they had, and they just tracked them over the entire, it was between like 30 months to 66 months. This specifically treats weak heart muscles. This is a very important distinction that you'll see why in a little bit, but it won't do things for fibrosis or other issues which happens when the extracellular matrix of cells gets too clogged up. It only helps treat them when they're like weak or dying. Questions? Okay, sorry. What they found, no significant benefits. This kind of sucks. They basically said that we have no statistical significant data to show that the primary endpoint wasn't met. And this means that they didn't see a reduction of recurrent heart events, so heart failures, heart attacks, all that. But, this is the big but here, they found that these mesenchymal precursor cells decrease the incidence of myocardial infarction, heart attack, and stroke. Oh, so 67% for heart attack, 56% by stroke. And they believe this was because the stem cells focus specifically on the anti-inflammatory properties, meaning it's only good for those weak heart cell muscles and not for the ones where the extracellular matrix makes them too hard. So it wouldn't fix all heart failure events, it only fixed about like half of them, which would just be these soft heart issues of stroke and heart attack. How does study fall short? So when you inject stem cells, they actually don't stay where you want them to stay most of the time. I found another study which shows that 1 hour after intracoronary injection, only 2 to 5% of the cells that were injected into the heart stayed there. The rest were in the liver and the spleen. So when 95% of the cells you're injecting don't even stay to do their job, it makes sense you wouldn't get a lot of benefits. We also didn't see a reduction in heart failure events. While stroke and heart attacks went down, the other issues did not. And despite looking at all heart failure events such as hypertrophy, which is another issue where it's the extracellular matrix, and fibrosis were not handled through these inflammatory pathways, and therefore saw no reduction, kind of making it to where the primary endpoint was not met at all. They also used the allogenic stem cells, which are from other people, not the own subject, meaning that they would have to be on immune suppressants for the rest of their life, and also it's just a lot riskier. There's a lot of issues here. Our capstone project, so this is a lot of words, but basically we want to integrate 2 different methods here. So we're going to continue with the transendocardial administration, which is the catheter through the inside of the heart, but we're going to also include epicardial patches. So these patches, if I go back to this one, are on the outside of the heart. They are either sutured or adhesed to the outside, and basically these are patches like band-aids made using stem cells. They have the extracellular matrix already created. They create that artificially, and then they grow the stem cells in there, making a little like sliver of heart that they can put on the outside of cells. This is a lot more invasive, which is why they don't use it all the time. But since you're already doing a transendocardial, which requires opening up the heart anyway, it makes sense that you could add it in addition. It also helps because they can find that, it also helps because a study found that the injection of the cells in addition to the patch has been noted to improve cardiac repair and maintain the amount of stem cells that stay in the heart. They found that the cell signals that the patch would send out keeps more of the injected stem cells within the heart instead of traveling through the vasculature into the liver and the spleen. Therefore, we're increasing more of the cells staying instead of just 5%. You could see way more than that. And it also just makes sense to do both of them because alone the patches have a problem where they won't adhere as correctly as they should to the outside of the heart, creating what you could consider just a scar on the outside of the heart with a little bit of heart muscle kind of floating by it. It also means that the patches don't beat at the same time as the regular heart muscles, meaning you're going to have an irregular heart rate, which, as we know, is not good. So if you do both of them, the injection will help electrically couple, which is the heart rate, the patches to the host heart, and then the patches will help the injections stay in the heart. So creating both of those will really kind of fix the issues that they both have separately. We also want to use autologous cells, which means that it comes from the person themselves, the subject. And since it's just bone marrow, it makes sense that you can just harvest it from themselves instead of having to have an entirely separate person. And this will also help curb inflammatory properties. And focusing strictly on the inflammatory aspect of heart failure events, so heart attacks and strokes, means that we won't be kind of bogged down by bad data of fibrosis and other extracellular matrix problems that come with the heart, that come with the heart side of heart muscles being an issue there. The goal, so these are the patches here, is to, we have a little two-part if-then statement for us. If autologous MPCs are duly transendocardially injected into the heart, so through the catheter, and applied to the epicardium via the patch, then stem cells will retain location within the heart and have increased integration and electrocoupling, meaning that they will fully kind of adhere to the heart muscle, eat at the same time, stay within the heart, and not go to the liver or the spleen and do God knows what. And then this will allow for neovascularization, so again, mini veins, which will help bring down inflammation because more heart muscles will be getting more blood flow, more oxygen, more nutrients, and this will also help with the just further reduction of stroke and heart attack effects while allowing for long-term health benefits. Another problem with this study was after the 30 months, they didn't see as much of a reduction in heart attacks and strokes because these stem cells probably all either diffused or the ones that stayed didn't stay for very long or were just weak by the point that they were adhering fully. So the introduction of the patch and the injection will hopefully have a more long-term benefit for people suffering with these little heart health benefits. Some little ethical concerns we have, because it is a life-altering process that is very invasive, this could be very expensive. And poor people have heart failure events too, so this could lead to only exclusive numbers of people having access to care. And then, as I said earlier, if the patches are not integrated correctly, the scar tissue can cause heart failure or arrhythmia, the irregular heartbeats. So you want to make sure the adherents and the injections are in the same location so that we can have the best chance at integration. And then the experimental treatment, this could be dangerous. We do have proof that it works in rats, but we haven't done the human model yet. So determining the long-term viability of this study is very necessary. But the last study, the one we're building off of, was the largest study in heart muscle failure that they've done in general. So it makes sense to kind of continue along this idea of studying the heart and just doing patches that we've already seen, proving in humans separately and putting them together. Any questions? Do you have a question?
Yeah. So like, what would be your rationale behind why people would pick this treatment over stents, possibly the input of stents, because stents is a lot less invasive and.
So stents is more like a Band-Aid. It doesn't fully fix the problem. So while that does allow for things to be open and the inflammation to kind of be curved, if they have more heart failure events that are recurring, it can just be an issue where they happen so frequently, the stents won't be able to help. This kind of seeks to just reduce hospitalizations in general. So while it is more invasive at the beginning, the goal is for them in between the 2.5 to 5 year period to just have less heart events and less hospitalizations than people with stents would have.
I might have missed this, but how, for your capstone project, are you guys planning to isolate your stem cells to the heart and prevent that running away?
So that's why we're going to include the patches. So a previous study found that there was retention between when patches were applied with injections, so dually applications there, because the paracrine factors that these cell patches secrete just improve the retention rate from that 5% number.
Did it say by how much?
It did not say by how much.
Cool.
Any other questions?
That's terrific. We'll do two. Awesome. Thank you guys. Really good questions. Yeah, so good question. Let's keep them going. Just a little bit of an update. Sorry, just a reminder. I'm gonna use this form. It's a little bit to grade the person presenting, but I'm also grading your feedback to them. Right. This is your way of kind of starting to interact. And so make sure you guys are giving me some good comments on conditions. All right, next group.
I'm next.
I guess with the other, are you guys the heart group?
Yes. Sure.
Let's go stay in the heart group. Let's go.
It was the other one, my bad.
Is it?
It's a weird thing.
Yes, we're doing. Do you grade your? Is it on this computer? Yeah, it's on this guy right here. Okay. I think it's just.
Oh, no. Wait, how do I make it bigger?
Go to slideshow.
Slideshow. All right.
Right here.
Right here?
Yeah, there you go. Hit the space bar. Or arrow keys, but.
Okay, our project was engineer heart muscle allografts for the heart repair in primates and humans. Okay, so the stem cells that were used, so we had two subjects. First was the macaque, which is a monkey. They used induced pluripotent stem cell synthesis, and they got this from the skin and or the bone marrow from the monkeys. And they used a multitude of transcription factors to turn these cells back into stem cells. And they generated cardiomyocyte stem cells and stromal cells. And this process occurred with the transcription factors. And then these cells were differentiated into the cardiomyocyte and stromal cells. These proved to work in the monkey experiment, so they then generated induced pluripotent stem cells in adult donor cells using reprogramming techniques. So basically for the patient, they took stem cells, or they took cells from an umbilical cord, and they used blood, the blood from the umbilical cord, and they turned these into induced pluripotent stem cells by feeding them transcription factors in vitro, and then once they had this in vitro, the patients received the EHM patches from which formed hundreds of millions induced pluripotent stem cells and used cardiomyocyte and differentiated them into cardiomyocyte and stromal stem cells to reduce the number of heart transplants and failing hearts. And this fixes key challenges associated with transplants and allows for, instead of just taking a heart out, you just put a patch on the heart of the failing muscles of the heart. And yes, those are the stem cells that were used.
It's just so laggy. If you hit the arrow, it'll take a second.
Okay. Any questions?
Try it.
Sorry, I kind of butchered that.
Are you hitting the right arrow?
Yeah.
Gilman, there's a question.
Yes,
This is a proactive or reactive method.
this is a reactive. So like your heart, basically what happened was the heart was failing on the patient, and they used IPS to differentiate cardiomyocyte and stromal cells, which are like allow the heart to keep functioning, and you don't have to do the transplant. But then I'll get that, this slide will change. I'll talk about what happens later in the presentation or in the experiment. I don't know why it's not changing.
Hit escape real quick.
Escape.
Slideshow again? Maybe just try going back to slideshow. Now it's going, so hopefully go back to slideshow and then let's just sing.
Okay, so to determine and differentiate our stem cells, they use flow cytometry. Okay, so the cells were tagged with an antibody, and these cells were dropped through the hydrofluidic system and struck with a light, and the cells were then assigned a specific charge, and the detector analyzed the transmitters of light, which basically showed the dye to differentiate each stem cell. And once identified, the two deflection plates created a magnetic field, and the cells that were charged were then sorted into two different categories. And the flow cytometry was used to confirm cell purity in the cardiomyocyte and stromal cell differentiation from human and rhesus macaque induced pluripotent stem cells and engineered heart muscles. So RNA sequencing was then used to analyze the cell composition and gene expression profile in order to clearly identify if the cells were going to achieve the desired result, which is engineered heart muscle cells, to make sure they'll function correctly. Are you getting it? Any questions? Well,
Clicking doesn't work.
That's crazy. Sorry.
Refresh the page. Computer just doesn't like you.
and
Sorry, you want to hit escape again.
oh, just okay, that's fine. No, it's working.
Just
This looks like AT's touch.
This is just a picture of how it would work. These would be the induced pluripotent stem cells, and then they shine a light through them. And then it tags the fluorescent antibodies, and then differentiates from positive and negative charge. Cool. So the main findings of the paper were it confirmed the feasibility of induced engineered heart muscle allografts in the macaque model, the reprogramming from skin fibroblasts is how they induced their stem cells. Long-term follow-up, so the stem cells in the macaque's heart lasted for 3 to 6 months, which proved that they were working. And then in the macaque, the heart wall grew from 4.5 to 0.6 millimeters in thickening, and no observed arrhythmia or tumor formation in the engineered heart muscles in the monkey macaque. I'm not sure if I'm pronouncing it. In the human trial, the heart muscles grafts were implanted in the human patient with advanced heart failures, and this is the trial BioBat HF phase 1/2, and explanted and observed with long-term follow-up. So they basically took the heart out from the human after because the heart didn't keep failing, but the patient wasn't getting much better. So they ended up doing a whole heart transplant. And here is basically the patch on the heart of the cardiomyocyte and stromal cells, which showed that they worked and they were able to continue the heart function. And then, yeah, they stayed in place and integrated, observed fewer capillaries in transplanted tissue, which basically means there's less little veins within the heart tissue from the integrated stem cells. And there was a mild immune response noted, but the patient was on immunosuppressants while the stem cells were in the heart to basically integrate the stem cells into the heart and immune response. Significance, the engineered heart muscle implants can survive and integrate into the human heart, and potential usage in advanced heart failure and try to prevent future heart failure. Let's see what that is. Any questions?
In the first article, they talked about the arrhythmia. Did that get mentioned at all with y'all's as well for the patch, like with the patch causing different
Yes.
heart rhythm?
So there weren't any confirmed arrhythmias in the
Oh, it looks like it says it right there, my bad.
Yeah. And then in the human, there weren't many arrhythmias. I can't remember exactly why they pulled the heart out. But
Looks like he might have just died.
For the human,
for the human,
that's what I was gonna ask, because if I remember correctly, what do you guys remember about that? What did they say why they ended the study with the human?
It wasn't working to their satisfaction, and she needed a heart transplant. And so, I think that also added to their ability to document some of the data.
Yeah. I don't know why in my notes, I was under the impression that I wrote that it was that because the transplant became available, they had to end it. Maybe I read it wrong. So I didn't know if it was a heart issue, maybe, but I could be mixing it up. Well, okay, you guys look into that one.
You're beyond immunosuppression.
okay, so for our capstone project, we want to use embryonic stem cells from a surrogate mother, probably, and direct differentiation of an embryonic heart valve development with valvular and interstitial cells, which are the majority of valve cells right here. Not the lining though, the endothelial is the valve lining. And then we propose that we create a 3D structure imitating the valve shape. So basically make a 3D structure with these two types of cells to create a similar patch to what they did on the human. And implant this into a child, and this would avoid size up surgeries, which are common with aortic valves right here, and allow the developing valve to develop with the child, so not have to keep doing surgeries and extending and enlarging the valve. think that's it. The sources on the next slide. Go.
You gotta hit the escape.
You got one question.
Yes. Yes.
Why embryonic stem cells and not just induced
Because-
something else?
We were gonna use embryonic stem cells because you don't have to, well, you do have to.
Because they can differentiate into anything.
Well, yeah, but we can do that with induced pluripotent too. So we just thought, since we have 5 people in our group, it would be harder ethics for us to fight about. So we thought,
It's a valid reason.
any more questions?
Science would say no, but for the class,
Yeah.
perfect answer.
Thank you.
So Air, basically our paper was with you all's capstone. I don't know.
That's what I
I don't know if you
noticed.
understand, but it's literally the patch, just minus.
The injection.
Yeah. But it didn't really seem to work super well, but it was kind of
Top right,
bad.
profile icon.
Just because they ended up having to take it right out.
Yeah, I mean, it's probably good that they were able to get it.
Yeah.
Looking at the patch like that definitely does not sound great. As you look at it, it's like a little tumor bubble. Yeah.
We're just going into your Google now.
Oh, there it is. Go look at it. They changed the name on that Google.
They're just gonna let me know one change. Like today. Finally.
So proud of you.
Senior year.
Oh.
Alright, so ours was on human skeletal stem cells. So this article used, they took fetal samples and they took samples from 10 different sites, where stem cells are present in the skeletal system. And so they were trying to figure out what causes degradation in bone and, bone development and in reproduction. So they found that most skeletal diseases are caused by aging and the downregulation of these stem cells through different signaling pathways. So to find the cause of these diseases, they used scRNA sequencing to determine the genetic factors for these diseases. And then the methods used to treat these were gene activation to upregulate the SSCs that were downregulated by aging. And it also found that the pathways the pathway that was being inhibited was hedgehog signaling. And then the pathway that was inhibiting was bone morphogenic protein signaling. And so after they took the samples from the 10 different sites, they found 4 different skeletal subtypes, which include chondrogenic, which is your cartilage, your osteogenic, which is bone generation, stromal, which is the outer tissue of the bone, and then fibrogenic, which is the fibrostromal tissue. So then we have to talk about scRNA sequencing, which is single cell RNA sequencing. So in this process, you isolate a single cell, and then you lyse it. So you break open the membrane and remove all the contents inside of it. So you remove the RNA, and then you turn it into complementary DNA. And then you take that complementary DNA and then compare it to a database to figure out which type of cell you have. And so in the article that we chose, they used the 3 different methods. So SmartSeq2 is just pretty much isolating the cell that they were looking for and opening up, lysing it. And then index sorting is where they tagged or they identified it with surface markers that were on the cell. And the computational clustering is just sorting what they got from the signal markers or surface markers and sorting them into their groups. So the experimental treatment and findings, so they attempted to initiate cellular reproduction in the skeletal stem cells by stimulating the hedgehog pathway and also inhibiting the BMP pathway. Because the hedgehog pathway upregulates bone development and growth, and the BMP pathway downregulates bone growth. And so they found that the degradation of the human skeletal stem cells mainly caused by aging, which also means that there were changes to the two pathways that they found. And so for our capstone project, although periodontal disease is not your typical skeletal disease, the advanced stages of periodontal disease affect the jawbone and the teeth. So periodontal disease is obviously just gum disease, and it's the leading cause of tooth loss, and affects about 50% of adults in the US. And in the advanced stages of periodontal disease, there's extensive bone and tissue loss. And then the teeth become loose and you can lose teeth, and there's damage to your jaw bones. So we plan for our experiment, it's gonna be centered around single cell RNA sequencing. And with that, we're gonna be able to determine the root cause of periodontal disease and what stem cells are being inhibited. So it could either be the tissue or the bone that's being inhibited. And so we're gonna examine the 4 subtypes of the human skeletal stem cells to determine the main region that's affected. And so if we find that the cause of this degradation is this similar to other bone diseases like osteoporosis, we plan to be able to treat it with mesenchymal stem cells from the wisdom teeth. So obviously potential treatment, wisdom teeth contain a surplus of mesenchymal stem cells that are capable of regenerating tissue. And so with this experiment, we're hoping to use the extracted wisdom teeth stem cells and insert them back into the patient, which would hopefully be able to regenerate bone or tooth. And so this is important because the current methods of treatment only slow the disease progression instead of actually curing it or making it better. So the use of HSSCs and MSCs attack the root problems of that current methods lack, because common treatment for periodontal disease is just antibiotics or oral surgery to remove degrading tissue. And so the success with treating periodontal disease could lead to broader implications within orthopedics. And yeah.
So in terms of like bone, like structure and stuff, we have like osteoblasts, osteoclasts, whatever. How is this different than something that slows down osteoclasts, which are typically used to break down bone and like provide basis for like regrowth?
Good question. Maddie, you.
So can you repeat that question?
Yeah, so we have multiple kinds of bone cells. We have osteoclasts, osteoblasts, osteocytes, just a bunch of different kinds of stuff. Osteoclasts are the cells that are responsible for taking apart old bone and like making a basis for new bone to grow in. So in some theoretical literature, you could have something reduce those, and so there's less being taken away. How is this different than that?
The main thing we were doing here is, so the original study was based off the regulatory pathways that cause the degradation of the bone. But what we're changing is, rather than trying to alter like the genetic pathways, we're gonna basically skip that step and just replace it altogether. So instead, it's gonna be brand new MSC cells from our wisdom teeth, which is probably an ethical concern because you gotta take someone's tooth out to do all that. And we're just taking all the MSC stem cells, from there and replacing the current bone tissue. That might answer.
You were talking about using wisdom teeth stem cells to help the person with their gum problems. But what happens when they get gum problems later in life and they've already had their wisdom teeth removed? Do you just pull someone else's teeth?
That's the problem with it, is
Okay.
that's also a concern. People probably aren't gonna want their own wisdom teeth to be taken out for someone else to use.
Wisdom teeth haven't really been used at all through this treatment, so
Right.
it's never really been a concern, so.
Can I ask you like a quick question? Like, because periodontits, like the whole thing is like the gums go away and then that exposes the teeth to the bacterial. If this only fixes the teeth, aren't the gums still a problem?
Yes, so our main goal was just in
Okay.
like the very advanced stages of periodontitis, when you let it get terrible,
So
you're
this is-
breaking down the bone.
This is like in addition to like when they get like a gum graft, you would also
Yeah,
do this.
Exactly,
Okay.
yeah.
Wait, good questions. Right now you guys are asking some really heavy questions about.
Hit
like,
the
stuff, which
top
I think
left.
It's awesome. I don't want to stop it, but
No,
if you're
never mind.
answering
Do
the
what you
question, and you're like, hey, I'm not sure, it's okay. Um, By the way, I really like that you guys all have been bringing up alternative strategies. Right? So that's a big thing with the ethics consideration.
What alternatives are there before what should we do with grading ourselves?
Just do it after class. Just drop it off. Yeah, even if you present it, you still gotta grade your own performance. If you don't remember, don't worry, I recorded it, I can share it.
Oh, I'm not looking at that.
Okay, our research paper that we used, essentially studied how stem cell transplants can be used to help heal eye injuries. The stem cells used within the research paper were autologous adult stem cells that were derived from the limbus, which is the area in the eye that borders the cornea and the sclera. And the function of those stem cells are essentially to maintain and regenerate the corneal epithelium. And so when there is a dysfunction within those cells that like ultimately lead to a deficiency, the cornea can't repair itself and it loses its smooth, glossy surface that causes blurred to lost vision, pain or irritation, or even recurring erosions or ulcers. And this can be caused by many different things such as infections, burns, trauma during surgery, or even genetic diseases. But within the study, these stem cells were harvested from a patient's healthy eye, and then they were grown ex vivo in an amniotic membrane using a two-step culture system. And then they basically that's when they become the cultivated autologous limbal epithelial cells, or like the calyx. And then after being like tested for quality and safety, they were transplanted back into the damaged eye. And so with this, the ultimate goal was to restore the missing or damaged cells and regain corneal function and regain vision. So yeah, just to reiterate, the goal of this research was to heal these eye injuries using your healthy eye. So all of these patients had one affected eye, one healthy eye. And they were kind of aiming to replace cornea transplants because cornea transplants do not address the limbal stem cell deficiency that Avery was talking about. And
these stem cells, they never turn into anything other than limbal stem cells. Since they were taken from the other eye, we don't need to turn them into any other form of potency, so they were always monopotent. And then, like Avery also mentioned, the stem cells, after they were harvested, underwent a lot of different methods to assess viability and a lot of other things to make sure that these stem cells were okay to transplant and that they would be effective once transplanted. But one that we'll focus on that we talked about in class is flow cytometry. It went through a lot of steps, but mainly the important parts of this are that they were washed with PBS, both the control group and the stained group, to get rid of any additional proteins that would affect the results of this. And this was to assess cell proliferation potential to make sure that once the stem cells were implanted, that they would act as stem cells and they would create more cells to heal the injured eye.
The main findings from this study, so first they were successful in making a xenobiotic-free, serum-free, antibiotic-free two-step cell culture, which basically just means they didn't use any animal-derived materials to make the culture. So that way it avoids any contamination with possible animal proteins or pathogens. And so then they manufactured the calic grafts and they maintained the epithelial stem cell characteristics and viability. And there were no infections or complications that arose from the clinical trial after the treatment. And all individuals were obtained a stable ocular surface. So basically just the surface of their eye became healthy and better. And so this offers potential for a new and safe, consistent treatment of LSCD using the individual's own stem cells. So this when it's immune response and possible infection.
Okay, and there were some kind of technical errors that we thought could inspire further developments in this CALIA technology, including how 2 of the patients in the original study, like Miriam said, had to undergo corneal transplants after the trial. And as Stella emphasized, all the patients had unilateral chemical burns. And lastly, all 5 patients that underwent the biopsies of the donor eye took about 4 weeks to fully recover and to return to baseline vision. And so from these, we'd like to propose a few points of interest for further development, including the improvement of the CALIA technology to prevent post-trial corneal transplants entirely, and exploring alternative outlets for viable limbal stem cells. For example, could we create a library of tissue-resident donor stem cells available for those without healthy limbal cells or those who experience bilateral chemical burns, and to work with those. Or could we harvest another type of a somatic cell that is easier to access than limbal stem cells, such as a skin cell, and can we manipulate them to become our desired stem cell type? Using mechanisms like nuclear transfer or induced pluripotent stem cells that could potentially reduce healing time and donor shortages and address. And yeah.
Any questions?
So you mentioned previously that y'all are using monopotent stem cells, right? That's also super cool, love that y'all are the only people who can do that for this. Is there any actual benefit for regressing into a more potent state, or is that just kind of more potential for something to go wrong?
I think it's just to address the injury to the healthy eye. I mean, obviously things can go wrong, and 4 weeks is not really an ideal time period of healing. So if we could lead into just getting something if you just skin will heal much faster than your eye will. So looking into those techniques to see if we could better address these donor shortages and people without the healthy eye. That's really the important part of our capstone project. People without the healthy eye, this obviously won't work for them because this requires you to have healthy limbal stem cells to be transplanted into the other eye. So that's really just a possibility to help and broaden it to help people with 2 affected eyes.
$12.50 exactly.
What do you guys think, class-wise?
Good.